But I find it will be interesting to this lecture also to highlight the failure of the innate defense or innate defense overwhelming. So the evasion means escape of innate immune responses by pathogens as a survival strategy. It is actually the pathogen may evade the innate immune responses just as a survival strategy. They follow several ways to evade or to escape immune responses. So pathogens may grow within host cell, not seen by the immune system. They will be sequestered or hidden inside the cell, so they will not be seen by the immune system. And as I mentioned here, that some pathogens, when they enter the cell, they downregulate expression of major histocompatibility class 1. And for example, viruses, when they enter some viral protein will bind to major histocompatibility class 1, and this major histocompatibility class 1 will be expressed on the surface of the cell, and this is a human cell, actually self cell, and major histocompatibility class 1 when get expressed on the surface of the cell, also is expressed with or in association with viral antigen. So this viral antigen will be recognized as non-self, and this will produce cascade of events that end in killing of the virus. However, the viruses, when they do that and downregulated major histocompatibility class 1, this will be sensed by NK cell, and NK cell also has the capacity still to kill or to destruct the infected cell by the virus. So as I mentioned that these microorganisms, in order to evade effective immune and responses and neutralizing immune responses, they grow within the host, and maybe they get sequestered inside the host and will not be seen by the immune system, or they express some molecules very similar to those of host cells, so they will be seen by the immune system as self cell, and or may cover themselves by plasma or by host membrane molecules. And in some instances, for example, in Epstein-Barr virus and other viruses, cytomegaloviruses, they produce interleukin 10-like. And as you know that interleukin 10-like, interleukin 10 itself, it downregulates macrophages. And as you know that macrophages, they produce human necrosis factor alpha and interleukin number 1. And also they produce interleukin number 12. Interleukin number 12 is very important for activation of T helper 1, and activated T helper 1 will produce interferon gamma, which it has antiviral activity. So some viruses, they produce some materials very similar to cytokines, for example interleukin 10-like by Epstein-Barr virus, or in some instances the pathogens may change the molecules on their surface, and this is known as antigenic variation. So when the pathogen is under attack by the immune system, it will change the gene segments, and then this will result in formation of new protein, and so the immune system will see this protein as if they see it for the first time, so it will produce a new defense against it, and then the pathogen will again change the service molecule, and this actual process may exhaust the immune system, and also the immune system in such condition may produce some antibodies of low affinity. Thank you very much, Professor Ahmed Bola.